Hi, today I'd like to share some of our group's recent work in using molecular dynamics simulation and machine learning to predict structures of cyclic peptides. So first, what are cyclic peptides and why do we care about them? Shown here is a diagram that has a drug modality. On the x-axis, we have the molecular weight of the drug modality. On the y-axis, we have the binding site surface area. So small molecules are something that we are most familiar with. For example, ibuprofen, Tylenol, and things that you can get over the counter. We said that these drugs have good pharmacokinetic properties. This means that we can take the drugs by mouth, which is very convenient for the patients. However, these drugs are called small molecules for a reason. Typically, they look like these little organic key molecules with molecular weight smaller than 500 Dalton. This means that these molecules are good at some things, but they are not good at everything. For example, they are good at targeting an enzyme binding pocket. However, if you want a drug that can target a large protein surface or interfere with the protein-protein interaction, then small molecules often do not have enough binding site surface area to do so. In these cases, you will look at a different type of drugs called biologics. These are antibodies and other proteins. They are large enough they can target protein surfaces with high affinity and selectivity. However, because they are proteins in nature, you cannot just eat them because they're going to get digested by enzymes. Therefore, you often see that cancer patients and other patients that they will need to do injections, which is very inconvenient for the patients. Furthermore, these molecules are so large they are unable to cross cellular membranes. This means that if your targets are intracellular, these molecules are unable to reach them. Now you might be wondering, are there things in the middle? And that is where cyclic peptides and other macrocycles are. It turns out that in nature, there are these fungi and bacteria that will make these cyclic peptide-looking molecules. Scientists have found out that they can be orally bioavailable like small molecules. They also can target large protein surfaces like biologics. So maybe cyclic peptides combine the best attributes of small molecules and biologics. However, if you are a pessimistic person, you might think, well, why do they have to combine the best attributes of the two worlds? Why don't they combine the worst attributes of the two worlds? If we look at the drugs currently on the market, we could see successful cyclic peptide examples. However, if we look further, we will find that most, if not all of them, are natural products themselves or derivatives of natural products. If we look at the drug bank, we will see that there are more than 2,500 small molecule drugs, more than 1,500 biologics, but there are very few cyclic peptide examples. This means that this concept is great. However, actually executing the concept and developing cyclic peptide that can, for example, target a protein that you are interested in, de novo seems very difficult. So what our group has been trying to solve is something that we call a structural challenge of cyclic peptide design. And I'm going to try to explain it using protein-protein interactions as an example. In each of these pictures, red is a huge protein, blue is another huge protein. They come into contact, trigger some conformational changes, the signal gets processed. These protein-protein interactions are very prevalent in biology, and specific protein-protein interaction are usually related to specific diseases. Therefore, we are very interested in being able to target specific protein-protein interactions as a treatment for specific diseases. If we have structural information, it's usually easier to design an inhibitor. If we look at all the protein-protein interactions that have been deposited into the protein data bank, we will see that more than 50% of the time, the interface residues adopt what we call non-regular secondary structure. For example, it looks like the protein on the left here is using some kind of loop-like structure, and the protein on the right here also use this little tiny loop to glue to the target gray surface. These are what we call hot loops. These are little loop-like structures that contribute a lot to the binding affinity. That's why they are called hot. Then today, if you, for example, ask a chemist, hey, I see this blue protein interacting with this gray protein using these four important residues, serine glutamate, as shown on the picture, in the picture on the right. Uh, what would you do if I need you to design a molecule? They will glue to the gray surface so that the blue protein can no longer bind to the gray surface. Then most chemists will likely say, hey, I want to synthesize a little short peptide that has exactly those four important residues that blue protein uses to glue to the gray surface. I want to mimic what the blue protein does. So you will synthesize a short peptide that has serine glutamate. However, chemists also know that if you have a short tetrapeptide, it's going to be so floppy in the solution that it's not going to want to adopt a single conformation that looks like that and glue to the gray surface. What the chemists come up with is an idea called pre-organization. I can synthesize these four important residues, then perhaps I can add a couple more amino acids represented by this red dash line. I can do a head-to-tail cyclization, for example. Then now I have a cyclic peptide. The important thing is I want this part of the cyclic peptide to look exactly like that. How can I best design the linker, which is represented by this magical red dash line in the PowerPoint, to best stabilize the desired loop structure? Today, if we know how sequence controls the structure of a cyclic peptide, then we can help solve this problem. Because if you design linker sequence number 1, then I am able to tell you that this is what the resulting cyclic peptide looks like. Then you can go ahead and see, oh, does this position the four important residues correctly? If so, this should be a good candidate. If not, even if you still have the four important residues, they are now pointing in some random directions, not effective at conferring binding toward this gray surface. Why are we not doing so at this moment? It is difficult to establish the sequence structure relationship for cyclic peptides. Cyclic peptides typically are between 5 and 15 amino acids, and you cyclize them in some way. So unlike large, well-behaved linear proteins, they are unable to adopt a cool fold like a alpha helix and beta sheet. When you throw cyclic peptides into solution, typically they tend to form multiple conformations at the same time. So you would have many different conformations. Each conformation has some kind of population. So you actually have a structural ensemble. This means that even if an AMR spectroscopist is willing to do solution AMR, the signal looks like a mess. They couldn't actually tease out easily how many conformations are there and what's the population associated with each conformation. This means that at this moment, it's very difficult to do structure-based design of cyclic peptides because we don't know how the sequence controls the structural ensemble cyclic peptides will adopt. Then now comes the second part of the talk, which is about molecular dynamic simulation. We just mentioned that it is difficult to characterize a structural ensemble of a cyclic peptide using solution AMR. However, there is a simulation technique called molecular dynamic simulation. If you have a sequence that you are interested in, say sequence number 1, a computational chemist is able to start a simulation of your desired peptide sequence. And at the beginning of the day, it might look like conformation A, but the simulation is going to run, and then your peptide is going to change conformation. By the end of the time, then we can come back and see, oh, throughout the day, what are the conformations your cyclic peptide adopts? Then we can come back and report to you that, oh, about 49% of the time, the peptide looks like this, 36% of the time it looks like this, and so on and so forth. Then you can use this structural ensemble information to do structure-based design. We can go over each conformation. Does the first conformation position the four residues correctly? Oh, it doesn't. The second one doesn't. Oh, the third one looks right. This is a great cyclic peptide structure that I want. However, the population is only 2%. Is it possible for us to tweak the linker sequence so that we can improve the population of this desired structure? Now I want to talk about several challenges in simulating cyclic peptides. So first is that unlike large, well-behaved linear proteins, cyclic peptides usually don't form alpha helix and beta sheet, which satisfy intramolecular hydrogen bonds using their backbone amide groups. Cyclic peptides typically have many solvent-exposed backbone CONH groups, and in extreme cases, you might even see cage water molecules inside cyclic peptides with high residence time. To a computational chemist, this means that it is important to describe the water interaction using explicit solvent simulation to capture the thermodynamics correctly. So to a computational chemist, if you say today that water is not important, then when I run the simulation, I can do something that looks like the box on the right. I will only have my peptide molecule, then I will treat water molecules as an implicit solvent, which means that if I have two partial charges interacting with each other, I just dial down the interaction by about 80. Imagining that I have water molecules, some kind of medium in the middle. But today, if you say that no, water molecules are very important, I want you to actually have explicit water molecules in your simulation, which means that I need you to fill the simulation box using HOH molecules. Then your simulation will look like the system on the left. The difference is the number of atoms in the simulation on the right is about 110. The number of atoms in the simulation on the left is 3,000. This means that the simulations run very slowly if you use explicit solvent simulation. A second challenge that we encounter is that it is difficult to sample conformational space of cyclic peptides, especially on the simulation timescale. So here is a movie showing a molecular dynamic simulation of a cyclic octapeptide that has 8 amino acids. I colored the backbone in light green, so this is like not a very exciting movie. If I now plot the 5' dihedrals in red and blue for residues 1, 2, 3, 4, 5, all the way to residue 8 as a function of simulation time. So I actually spent about 5 days running this simulation, watching the peptide do almost nothing. So we could see that for the first 5 residues, the 5' dihedral angles almost did not change. This means that my cyclic peptide is actually stuck in a local fringe minimum, and its conformation is not being sampled well. I can easily start with a different initial structure, then I see similar thing that my peptide tends to be stuck in a specific conformation. Once in a while, it will change conformation and then continue getting stuck. If I start with 2 initial structures, by the end of the day, I could not get very similar results. This means that I have a sampling problem, and I'm actually now ready to tell you what this cyclic peptide actually does regarding its conformations. So we have a sampling problem. To kind of drive the message home, this is a Raman-Chandra plot, which is a 5-site density distribution for residues 1, 2, 3, 4, all the way to residue 8. The green dot is the 5-site dihedral angle values each residue has when you first start the simulation. The green cloud little dots are where the peptide samples the backbone dihedrals during the simulation. So you could see that it quickly falls into some local minimum. So except maybe like residues 6, 7, and 8, residue 1 to 5 do not actually sample other regions. Then when you start your simulation in a different structure in simulation number 2, then what we saw is the areas that you sample do not overlap. However, if you have good uh, converge simulation results, then regardless of where you start your simulation, you should actually be able to report similar structural information. What we have here is a sampling problem. So conceptually, if say you have a free energy profile that looks like this, you don't know what it looks like. That's why you are running a simulation to see where where your cyclic peptide likes the best, where your cyclic peptide likes the second, and so on and so forth. However, if you start your simulation over here and this free energy barrier is very high for your simulation to cross or your system to cross during the simulation time at this temperature, then what you will see is as I run the simulation, I see that this peptide is going nowhere. So this is a signal that you are getting stuck in a local free energy minimum. So how can we solve this problem? So what we're going to use is something called metadynamic simulation. So in metadynamic simulation, meta means change. So we're going to push the system away from local free energy minimum by depositing repulsive Gaussian potential along a specific coordinate. What this means is, for example, the underlying free energy profile again looks like this. You start your simulation and your peptide really likes the value 60. So now you're going to take a notebook, then you're going to say, oh, you really like 60. Let me make you hate 60 by depositing this repulsive Gaussian potential in your simulation. Then your peptide is going to say, okay, fine, I like 75. Then you write down, okay, you like 75. Let me make you hate 75. So you play this game with your system that many times, it's going to say, fine, I don't like the 60-ish angle value anymore. Let me do minus 90. So we repeat this exercise until your system now traverses minus 180 to 180 evenly. So you enhance the sampling of your cyclic peptide, of your peptide. However, what you want to do is, well, without me messing with you, what do you like the best? Then you take your notebook, you add up all those repulsive Gaussian potentials that you added to your system, you will have a very intriguing shape. This must mean that to make the peptide hate or love all the places equally, you really need to make it hate here, right? So if I invert this curve, it actually provides the original underlying free energy profile without the biases that we added to the system. And this is how we can recover the equilibrium free energy profile using this method. There is one challenge of this enhanced sampling method that is you need to select a good coordinate for the algorithm to bias. So you need to tell the algorithm, hey, what I need you to do is, bring the two termini together closer, or I need you to rotate this dihedral angle, or I need you to bring these two things together, or what do you actually need the computer to do to help your system sample different conformations? Now I am in a bind because the challenge I had is I did not see my cyclic peptide move. Therefore, I need this algorithm to help me move my cyclic peptide. But for it to do so, I need to now tell the algorithm what my cyclic peptide needs to do to change conformations. This is like a catch-22. To address this problem, we now need to understand how cyclic peptides switch conformations so we can tell this algorithm, hey, this is the specific motion I need you to help me with. To provide insights into how cyclic peptides switch conformations, we run a molecular dynamic simulation of a very specific sequence, cyclohexaglycine, for example. We intentionally use glycine because it's the smallest and hopefully the most flexible cyclic peptide. We expect this sequence might be relatively flexible. So if we run a molecular dynamic simulation, perhaps we can actually see many conformational switches. So we can analyze what a cyclic peptide needs to do when it switches conformations. Here on the left, there's going to be a movie. On the top is the structure of the cyclic peptide. On the bottom, this bar graph is going to tell us at this moment which residues are forming a beta turn and what type it is. 6 is a great number for cyclic peptides because two residues typically form a beta turn, and two residues form another beta turn, and the two residues in the middle can potentially form two hydrogen bonds. There are four types of beta turns people typically discuss, so I'm going to color them differently. At this moment, residues 3 and 4 are forming a type 2 green beta turn. So as the simulation starts, what we are hoping for is that please let me see conformational switches, right? So maybe this peptide is flexible enough that I can actually observe conformational changes so I can try to understand what cyclic peptide does to switch conformation. So some things happen, right? So something happened that this cyclic peptide actually switched conformations. So we analyzed many instances of such conformational switches, and we found out that there are two major switching mechanisms. In the first case, for example, at this critical moment, two residues switch from a type 1 beta turn to a type 2 prime beta turn. If I now plot how much phi 1 psi 1, phi 2 psi 2, all the way to phi 6 psi 6 change from one moment to the next moment. If this dihedral angle changes value by a lot, I will color it red. If it didn't change a lot, I will color it white. We see that at this important moment, there are two dihedral angles, phi 1, psi 1. They both change by more than 90 degrees at the same time. When I saw the result, I'm like, yeah, because it is a cyclic peptide. If we have a linear peptide, we can drag one dihedral angle by more than 90 degrees. It's fine. But if you have a cyclic peptide, you cannot just drag one dihedral angle by 90 degrees because you're going to break the ring. So you need two dihedral angles to play hand in hand. We found out that the two dihedral angles are pretty local. So typically the phi and psi of the same residue, or the psi of one residue and the phi of the next residue. This allows us to go back and tell our metadynamics simulation that, hey, for this rigid cyclic peptide, what I need you to help me do is help me make these concerted two dihedral angle motions so that I can sample the cyclic peptide conformations efficiently. This is to compare if you run 1 nanosecond of conventional MD, then we can see that the backbone is basically locked. You are not actually sampling other conformations. But if you run 1 nanosecond of this bias exchange metadynamic simulation, we can efficiently sample different conformations in the metadynamic simulation. So what we can now provide is if you start with two different initial structures, then we kind of guarantee that, by the end of the simulation, your structural ensemble results are very similar. So typically for small cyclic peptide, we can achieve that using about 100 nanoseconds of simulation, and that takes around 1 day of human time. Using molecular dynamic simulations with this enhanced sampling method, we intentionally design well-structured cyclic peptides so that the AMR signals will look simple, clean. Our experimental colleagues can hopefully deduce the structure and then compare to the simulation prediction. So I'm not going to say that we are 100% correct, but I think we are about 70% correct, which means that if we predict this cyclic peptide sequence to be well structured, and then our experimental colleagues synthesize the peptide and does a solution AMR, the AMR restraints and dihedral angle values deduced are going to be reasonably consistent with the simulation prediction about 70% of the time. Now can we do what we set out to do? So here is an example. A colleague is interested in designing a cyclic peptide that can target this protein called KEAP1. We know that there is a protein called NERV2 colored in green here that uses these three important residues to engage KEAP1. Then the idea is, can we actually design a cyclic peptide that mimics these three important residues that position them correctly to bind to KEAP1 efficiently? We will run simulations on many different designs with different linkers, different sequences. Then we watch the cyclic peptide only, and we calculate one thing. That is, what percentage of time I see that the cyclic peptide positioning the three important residues correctly. So we call that percent pre-organization. Your intuition tells you that you probably want this number to be as high as possible. Using this metric as a guide, we can actually help our colleague to design cyclic peptide from, for example, this is a micromolar binder to guide it all the way to a nanomolar binder. So hopefully, I have now shared with you how molecular dynamic simulation can be a helpful tool to provide structural information of cyclic peptides. Then why the third part, machine learning? So what we have right now is, we have a great technique, molecular dynamic simulation, that can provide this like missing piece in the puzzle, the structural information to allow people to do structure-based design. But am I ready to, go talk to like a big pharma company or biotech company and say, hey, I can predict cyclic peptide structure. Let me help you, with your screening effort. So they're gonna probably ask me, how many sequences can you simulate in a day, then I will have to tell them that even if I use all the computers that my group has, I can probably run such simulations for 31 sequences in a day. Right? They're not going to say, the door is that way, because they want you to be able to provide structural prediction results for cyclic peptides, probably, thousands a day or tens of thousands a day, because they want to be able to screen many different sequences very quickly. So the challenge at hand is, I have a good technique, but it is a slow technique. So how can I turn a good but slow technique into a good and fast technique? Then this is when the machine learning comes in. So by now I have simulated a lot of cyclic peptides. Why don't I take my simulation results and use a machine learning model to recapitulate those simulation results? Therefore, when you ask me to run a simulation of a cyclic peptide, I probably say, let me just ask this machine learning model what structures and population this cyclic peptide would have, as if I actually run the simulation. The idea is, well, there's no free lunch. You have to bite the bullet once. So run molecular dynamic simulation of all these sequences. So the lowercase letters here are D amino acid. Then analyze the structural ensemble results. For example, when the sequence is SNSVA, I see you have so many different structures. The population for the first structure is 2.27%. For second structure is 0. Third structure is 3.22%, so on and so forth. Then when I have different sequences, then they have different preferences for different structures. So for each of this row, the numbers will sum up to 100%. As you change the sequences, you are changing the distribution of numbers among these various structures. Then once I have those results from simulation, I'm going to build a machine learning model to recapitulate this simulation results, meaning that if I input SNSVA, I want the machine learning model to output 2.27, 0, 3.22, and so on and so forth. The first thing we need to do is to actually encode structural ensemble in a way that a model can understand or accept. We start with cyclic pentapeptides as our first study. We focus on the backbone conformation. We run a simulation of cyclic pentaglycine, as a glycine is the smallest, most flexible cyclic peptide, trying to get an idea of what conformations cyclic peptides can adopt. Here is a density distribution of cyclic pentaglycine. So we could see that there are, some bi-side dihedral angles the cyclic pentapeptide likes. So using this density distribution as a guide, we could separate the whole 360 by 360 area into 10 different regions. Each region is given a structural digit coded by a Greek letter. Then when I run a simulation of a pentapeptide of any sequence, then I can look at a structure. I can measure the 5-side dihedral angles of each residue. Then use my map, I can actually encode a structure using 5 structural digits. For example, I can look at the first amino acid, all the 5' angles are here, then they fall into the pink area in my map. So I'm going to code this structural digit lambda. The second one is beta, third one, so on and so forth. So by measuring the dihedral angles for each amino acid, I am now encoding the structure of a conformation using a string of 5 structural digits. Then now I can look at all the different conformations during the simulation, measure the dihedral angles, give it a structural digit string, and count how many unique structural digit strings I have seen in the simulation and what's the population of each structural digit string. How do we design the training data set? So what we want to know is the population of an amino acid sequence X12345 adopting structural digit 12345. Then what we think might be important is, like the sequence, right? Like the nearby sequence, what is, first two residue, is it GG, GA, or AA, and so on and so forth. So very talented postdoc with a very strong physics background think that, okay, maybe we should design like a set of sequences that we promise that we will actually observe all possible X123 tripeptide patterns. So then we run simulation of all these 705 random, semi-random sequences. Then we analyze the simulation results. Then we build a machine learning model. So here is a result. So we call this machine learning model like a StrEAMM model. So StrEAMM stands for Structural Ensembles Achieved by Molecular Dynamics and Machine Learning. So for each of these dots, it represents what for a specific sequence, a cyclic peptide sequence, adapting a specific structure, what is the population? So on the y-axis is what you saw in the molecular dynamics simulation, so everything is smaller than 100%. On the x-axis is what the model fits to. So you want y equals to x. And this looks pretty good, but this is for the training data set. Now, if I actually give the machine learning model a new sequence, can you make good prediction? So now if I now input SVFAA in less than 1 second, the machine learning model is going to tell you this is a top structure population, second structure population, and so on and so forth. If you spend 1 day to run the simulation, you're going to get the top row. So, like I have standard, but this looks pretty good. We test this on 15 new random sequences, and the R-square is about 0.96. This means that instead of using all the computer resources that my group has to be able to run simulations for 31 sequencings a day, now we can use the machine learning model to actually output more than 10 million predictions using 1 CPU in a day. So for the last part, I want to kind of share some outlook. So our group is very interested in extending this StrEAMM platform into other cyclic peptide scaffolds. So I always learn a lot by collaborating with people. I, as a computational chemist, really like head-to-tail cyclized peptides because your whole system is still peptide in nature, so you don't need new parameters. However, we have had colleagues, they complain to us that, Yu-Shan, can you stop doing head-to-tail cyclized peptides? I'm like, why? It's like, because the chemistry is not like robust, meaning that the yield sometimes can be very low because I need to synthesize a linear peptide and hoping the two ends will actually come together so that the peptide can cyclize. And then for a short peptide, how likely is it that the two ends are going to come together, right? So sometimes the yield is so low they couldn't even synthesize the peptide for us. And we are also aware that a lot of labs and companies use things like mRNA display to screen for cyclic peptides. Then typically, you kind of need like a hook, which is like your terminus to actually connect the cyclic peptide to a screening platform. Then if you do head-to-tail cyclized peptide, what will happen is, well, you don't have a terminus left. So typically, it is not compatible with mRNA display. So our group is also very interested in extending this simulation machine learning structural prediction platform to other cyclic peptide scaffolds, including the thiol ether scaffold or other cyclization methods via sidechain cyclization. We're also very interested in incorporating non-natural amino acids as one of the typical kiss of death for peptide therapeutics is that they get degraded by enzymes too fast. So we want to throw in things that are unnatural so that the enzymes don't recognize them easily. And incorporating different types of amino acid also allow you to have a larger toolbox to actually tune different properties. So I want to mention that our recent work in being able to incorporate more amino acids in the library with almost like no additional cost. So if you look at the slides very intensively, and you might notice that hey, in your study you have D and L amino acid both, but your library is kind of small. You pick like 15 representative amino acid. Like, can you potentially use your model that's trained using 15 amino acid and applied it to make predictions of cyclic peptides that are, from a library of, 50 amino acid instead? We encode amino acid in our model using, for example, molecular fingerprints. So this means that each amino acid is going to be represented by a barcode of zeros and ones. So you have 2048 zeros and ones in a very simplified version to explain this, then what each bit represents if you have a specific functional group. For example, the first bit represents do you have a CH3? The second bit represent, do you have a CO? Third bit represent you have a NH2 or so on and so forth. Then if your molecule has that functional group, then the bit will be turned around into 1. If your molecule does not have that functional group, then the bit will be 0. So then each amino acid can be represented by 2048 zeros and 1. And for example, glycine being the simplest amino acid, it's going to have a lot of bits being 0. Arginine, alanine, or other more and more complicated amino acid going to have more bits getting turned on to 1. So I can actually encode all amino acids, including, for example, your non-natural amino acid on the same footing. So I can use my model to make the prediction in the sense that the computer is not going to explode. But is the performance good? If I train my model using a 15 amino acid library, when I test the model on cyclic peptides composed of the same 15 amino acid library, the R-square is very good as we saw before. However, if I use this model to make predictions of cyclic peptides, they are made of a larger amino acid library, then you can see the performance is rather poor. So how do we actually allow the machine learning model to extrapolate to new amino acids better? The idea we came up with is, well, if you have an amino acid you're interested in, what I would do is I will actually run a simulation of a dipeptide of your amino acid, which means I'm going to take your amino acid, I'm going to cap it on the N and C terminus. I will run the simulation of that simple system. What it allows me to get is the phi-side dihedral angle preferences of your amino acid. Different amino acids are going to have different preferences. For example, glycine is going to be able to sample both positive and negative phi dihedral angles. And this is L-alanine. When it's D, it prefers positive phi values. When it's valine, it's going to have different features in the preferences of the phi side backbone dihedrals. So by running a short simulation of dipeptide, then we can actually categorize the preferences of backbone dihedral angles for specific amino acids. Then we can actually use this as an input for our machine learning model. So each amino acid is going to be represented by the backbone density distribution that is sampled during a dipeptide simulation. Then now if instead of using molecular fingerprint to represent your amino acid, if you use this backbone feature to represent your amino acid, what you could see is you get much better free lunch. Like your model that's trained on using 15 amino acid now can be extended to new amino acids more accurately. So hopefully today that I share with you how we can use simulation and machine learning to provide high quality structural predictions for cyclic peptides. I just want to say that we are also very interested in using simulation and machine learning to be able to predict properties, for example, binding affinity and membrane permeability. We are in active collaborations with both experimental groups in academia and in industry. I'm glad to have the opportunity to share with you what our group has been doing recently using simulation machine learning to be able to help people better design cyclic peptides. And I'd like to thank all my group members and collaborators, the funding agencies, and well, thank you so much. I hope that you now know cyclic peptides and the simulation machine learning approach a little bit better.